Recently, we have developed a Omicron-specific qPCR assay for detecting which positive COVID patients have the Omicron variant compared to not having the Omicron variant. Over the course of the pandemic, our lab and the public health department and the physicians in Santa Barbara have formed like a really close network for distributing information and sharing what's actually happening, whether it's in the hospitals or on the research side of things. And this network that we've created has allowed us to strengthen the role research plays directly in clinical applications here in Santa Barbara. The clinicians reached out to me and the members of our lab and said, hey, we need a way of detecting Omicron and we need to be able to do it very, very fast because we think it's spreading to our community and we need to adjust the way we're treating our patients if it is. The reason we developed this assay was because the typical way of determining what variant a patient has required next-gen sequencing of the entire SARS CoV genome from that patient. While this process gives us a ton of information, it's unfortunately slow and can be costly to get a large amount of data. For those reasons, we're not able to screen every positive that the county sees for, do we have this variant or do we not have this variant? So knowing which variants are spreading rapidly through our community provides at the high level guidance to our clinicians of what they should do with their patients. If you know you have a treatment that only responds to one variant and not the other, and you know that that variant is not present in our community or is massively present in our community, they can have more comfort deciding what to treat their patients with. We plan to release the method behind this assay and all the specifics of the design on like a preprint very soon, the writings in the process, in order to share it with the general scientific community and to kind of use it as an example of how one single lab can help protect an entire county via just some small assay development work, and really emphasize that working closely with our physicians and our public health department can have really critical, beneficial outcomes to public safety. So now that we have a system for designing these tests, we can implement that for the next variant. So let's say we see something that's not Omicron starting to take hold here in Santa Barbara. What we can do is target those samples, sequence them, figure out what the genome is of these non-Omicron samples, find the mutations in them, and just redesign the assay to target those mutations. That gives us a platform for quickly transferring to the next variant and hopefully catching these outbreaks almost immediately and preventing them.